Good morning, I'm Joanna Eliasol Vicente, and I'm here to talk about carbohydrates metabolism. To start, carbohydrates are sugar molecules. Along with proteins and fats, carbohydrates are one of three main nutrients found in foods and drinks. Carbohydrates are probably the most abundant and widespread organic substances in nature, and they are essential constituents of all living things. Carbohydrates serve as energy sources and as essential structural components in organisms. In addition, part of the structure of nucleic acids, which contain genetic information, consists of carbohydrates. Carbohydrates metabolism, on the other hand, is a fundamental biochemical process that ensures a constant supply of energy to living cells. The chemical makeup of carbohydrates. Carbohydrate is a biomolecule consisting of carbon, hydrogen, and oxygen atoms, usually with a hydrogen-oxygen atom ratio of 1:2:1. Organic compounds are organized into ketones or aldehydes with numerous hydroxyl groups that are derived from the carbon chain. The general formula for carbohydrates is CxH2OY. The carbohydrates are structurally represented in three forms, and these are open chain structure, hemiacetal structure, and Haworth structure. There are numerous functions of carbohydrates. In living organisms, they make use of carbohydrates for energy that can be used to fuel cell reactions. They are the biggest food source of energy in all living things. Carbohydrates, in addition to being the main energy source in a variety of animals, are a quick source of energy. The breakdown of glucose is accomplished by Krebs cycle and glycolysis to create ATP. It also serves as energy stores as well as fuels and metabolic intermediates. Glycogen is stored in animals and starch in plants. Stored carbohydrates are the source of energy and not proteins nor fat. They are structures and protectants such as in the cell walls of microorganisms as well as plants. Structural elements found in the wall of the cells in microorganisms and in plants as well as in mammals is also one of the functions of carbohydrates. Carbohydrates are immediate in biosynthesis of proteins and fats. They help regulate the function of nerve tissue and are the primary energy source for the brain. Carbohydrates are linked to proteins and lipids, forming receptor molecules, antigens on the surface, as well as vitamins and antibiotics. It is also responsible for the creation of the structure of the RNA and DNA. They are connected to numerous proteins and lipids, and these linked carbohydrates are crucial for cell-to-cell communication. And in animals, they are an important component of connective tissue. Having a high fiber content can help in preventing constipation, and they also aid in regulation of the immune system. Carbohydrates provide calories and energy. It can be in the form of simple carbohydrates, which are ring-like molecules made up of carbon, hydrogen, and oxygen, either alone or in pairs. It is mixed in foods to sweeten, prevent spoilage, or improve structure and texture, like fueling the yeast in doughs. Complex carbohydrates, which are formed when these rings are hooked up together. It is composed of three or more monosaccharide units strung together in long complex chains, which are high in fiber and are digested slowly by the body. Examples are rice, pasta, soybeans, and lentils. Carbohydrates can be simple or complex. Simple carbohydrates can be monosaccharides and disaccharides. Examples of monosaccharides are glucose, such as honey, molasses, dried fruits, fruit juices, and sweet corn. Fructose can be found in fruits and even in soda drinks. And galactose, a monosaccharide that can be found in honey, yogurt, celery, basil, among others. Examples of disaccharides include maltose, which can be found in wheat, cornmeal, and in some fruits like pears and peaches. Lactose is a disaccharide that can be found in milk and dairy products. Sucrose is also a disaccharide that can be found in table sugar, sugarcane, and dates, to name a few. For complex carbohydrates, we have polysaccharides. Examples of this are starches found in bread, cereals, pasta, rice, and the likes. Another are fibers which can be found in brown rice, fruits, and vegetables. Glycogen is a polysaccharide. In humans, glycogen is made and stored primarily in the cells of the liver and skeletal muscle. These are structures of some carbohydrates. We have monosaccharides like glucose, fructose, and galactose. We have disaccharides, a combination of two monosaccharides like sucrose from glucose and fructose, and lactose from galactose and glucose. And polysaccharides coming from bonds of ring-like structures of carbohydrates. We have cellulose, starch, and glycogen. We will now continue with the digestive pathway of carbohydrates. Digestion of carbohydrates begins by breaking of polysaccharide into smaller saccharides aided by various enzymes. In the mouth, chewing increases surface area of food, allowing salivary alpha amylase to begin digestion of starch. Some of the free glucose in food can be absorbed through lining of the mouth. When carbohydrates reach the stomach, digestion halts temporarily. When it reaches the small intestine, the stomach contents are neutralized by pancreatic juice. Pancreatic amylase continues digestion of starch to oligosaccharides and maltose. Intestinal brush border membrane enzymes are the following: maltase, which hydrolyzes maltose to glucose; sucrase, which hydrolyzes sucrose to glucose plus fructose; and lactase, which hydrolyzes lactose to glucose plus galactose. The end product of carbohydrates digestion are glucose, which are readily absorbed through the intestinal mucosal cells into the bloodstream. Absorption of monosaccharides by intestinal mucosal cells. The different sugars have different mechanisms of absorption. Glucose and galactose actively taken up by mucosal cells through energy-dependent secondary active transport. Fructose, on the other hand, is taken up by facilitative transport down concentration gradient via passive carrier-mediated transport. Absorbed sugars diffuse from intestinal mucosal cell into portal circulation and are transported to the liver. After being absorbed from the intestinal tract, the monosaccharides are carried by the portal circulation to the liver. It is the first organ to receive glucose, fructose, and galactose. The liver takes them up and converts galactose to glucose, breaks fructose into even smaller carbon-containing units, and either stores glucose as glycogen or exports it back to the bloodstream. Depending on the supply and demand, the process of breakdown includes glycolysis, glycogenesis, glycogenolysis, gluconeogenesis, and pentose phosphate pathway. In the large intestine, any carbohydrates that weren't digested in the small intestine, mainly fiber, pass into the large intestine, but there is no enzymatic digestion of these carbohydrates here. Instead, bacteria living in the large intestine, sometimes called our gut microbiota, ferment these carbohydrates to feed themselves. Moving on to the carbohydrate metabolism overview. Glucose is a simple sugar which is an important energy source in living organisms. It will be the source of ATP production. Depending on the body's demand for energy, glucose in excess will be stored as glycogen via the process of glycogenesis. When the body demands for energy, the stored glycogen will be broken down back to glucose through the process of glycogenolysis, and through glycolysis will be converted to pyruvate, then converted to acetyl-CoA, and enters the citric acid cycle to produce ATP. Glucose molecules can also be derived from other sugars via the process of gluconeogenesis. Pentose phosphate pathway is defined as a metabolic pathway that occurs in all living organisms, and it utilizes the first intermediate product of glycolysis. In the absence of oxygen, pyruvate is converted to lactate or ethyl alcohol in plants. In the glycogen formation, if glucose from carbohydrates digestion is in excess and forms excess blood sugar right after a meal, it will be stored as liver glycogen. The process in which glucose is being converted to glycogen is called glycogenesis. And in the process, there are different enzymes that assist in its conversion. These are hexokinase, which is coming from the muscle, the glucokinase, which is in the liver, phosphoglucomutase, UDP glucose pyrophosphatase, glycogen synthase, and branching enzymes. If glycogen is broken down to glucose in a series of reactions as the cells of the body use up the glucose in the blood, the process in which glycogen is being converted to glucose is called glycogenolysis. Glycogenolysis is actually may happen in the liver or in the skeletal muscle. The steps in glycogenolysis and end products differ based on the cell type. Both products, though, can join glycolysis. In the process of conversion or converting glycogen to glucose, the following enzymes are present. We have the glycogen phosphorylase and glycogen synthase. We now move on to glycolysis. Glycolysis is a cytoplasmic pathway which breaks down glucose into 2,3 carbon compounds and generates energy. Glucose is trapped by phosphorylation with the help of the enzyme hexokinase. Adenosine triphosphate or ATP is used in this reaction, and the product glucose 6-P inhibits hexokinase. Glycolysis takes place in 10 steps, 5 of which are in the preparatory phase and 5 are in the payoff phase. Phosphofructokinase is the rate-limiting enzyme. ATP is generated by substrate-level phosphorylation by high-energy compounds, such as 1,3-biphosphoglycerate and phosphoenol pyruvate. Glycolysis is used by all cells in the body for energy generation. The final product of glycolysis is pyruvate in aerobic settings and lactate in anaerobic conditions. Pyruvate enters the Krebs cycle for further energy production. Now we have gluconeogenesis pathway. It is the synthesis of new glucose from non-carbohydrate precursors. Provides glucose when dietary intake is insufficient or absent. Examples would be lactate, alanine, pyruvate, glycerol, glutamate, and other glucogenic amino acids. It also is essential in the regulation of acid-base balance, amino acid metabolism, and synthesis of carbohydrate-derived structural components. This is the catabolism of proteinogenic amino acids. Amino acids are classified according to the abilities of their products to enter gluconeogenesis. These classifications will include glucogenic, ketogenic, and both glucogenic and ketogenic. Glucogenic amino acids are amino acids that can be converted into glucose via gluconeogenesis, such as alanine, glycine, cysteine, serine, and the others. Ketogenic amino acids are amino acids that form acetyl-CoA or acetoacetyl-CoA. Examples would be lysine and leucine that can be converted to acetyl-CoA before it enters the Krebs cycle. Another classification is both glucogenic and ketogenic, such as tryptophan, tyrosine, phenylalanine, threonine, and isoleucine that can be converted to acetyl-CoA before it enters the Krebs cycle. The Cori cycle is a cyclic biochemical process that lactate can be transported in the blood to the liver where it is converted into glucose. It allows for reutilization of lactate or lactic acid produced during exercise and minimizes muscle cramps due to lactic acid buildup. We will now discuss the aerobic and anaerobic pathways and the production of ATP. In the aerobic pathway, the carbohydrates in the presence of oxygen will produce carbon dioxide and water. If the cell contains enough oxygen, the pyruvate from the glycolysis of glucose is oxidized to acetyl-CoA. The reaction requires enzymes and coenzymes and produces carbon dioxide and NADH. Okay, from glycolysis, the two pyruvate molecules will be converted to 2-acetyl-CoA, and it will enter in its form the Krebs cycle, or also known as the citric acid cycle or the TCA or tricarboxylic acid cycle. Inside the Krebs cycle, there are 8 chemical steps that require enzymes, and these are citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, L-malate, and oxaloacetate. Now, there are 2 reactions which are requiring water, and these are in this area and in this area. And there are 2 reactions which are carbon dioxide producing, here and here. There are 3 reactions that yields NADH and 1 gives FADH2. NADH is being yielded here and in this part, and FADH is in this part. And all of these reactions takes place in the mitochondrion. The electron transport chain. The electron transport chain is located within mitochondria, and the proteins of the electron transport chain span the inner mitochondrial membrane. The hydrogens in NADH and FADH2 are passed along intermediate hydrogen carriers, and the electrons transfer all the way to oxygen that will actually produce a more usable energy for metabolic use. Each NADH is equivalent to 3 ATP, and FADH2 is equivalent to 2 ATP. When the electrons are transferred to oxygen in addition to hydrogen, it will produce water. The enzymes and other components of the ETC are in the inner membrane of the mitochondrion. During the course of the electron transport chain, only 2 things are really created. First, water is created as the electron transport chain deposits spent electrons into new water molecules. These water molecules can be reabsorbed by the body for use elsewhere or can be expelled in the urine. Second, while the electron transport chain does not create ATP, it does create the proper conditions for ATP to be produced. This is called the proton motive force and is a product of the electron transport chain transporting hydrogen ions to one side of the inner mitochondrial membrane. One of the best ways to understand the function and purpose is to understand what happens if the electron transport chain stops. This can happen from 2 basic scenarios. The electron transport chain can stop because it does not have a source of electrons, or it can stop because it can no longer pass electrons on. The first scenario would be caused by something like starvation. In the process of aerobic pathway or respiration, there are 36 ATPs produced. Aerobic respiration typically produces a net total of 36 ATP per molecule of glucose consumed. A net total of 2 ATP are produced in glycolysis via substrate level phosphorylation. 4 are produced but 2 are consumed. A further 2 ATP are similarly produced in the Krebs cycle, 1 ATP per cycle. 2 cycles occur per glucose molecule. Lastly, 32 ATP are produced in the electron transport chain using energy from hydrogen carriers, or it's called oxidative phosphorylation. Hydrogen carriers produce different amounts of ATP depending on where they donate electrons to the transport chain. NADH molecules located in the matrix donate electrons to the start of the chain and produce 3 ATP per hydrogen carrier. Cytosolic NADH from glycolysis donate electrons later in the chain. and only produce 2 ATP per hydrogen carrier. FADH2 also donates electrons later in the chain and so only produce 2 ATP per hydrogen carrier. During oxidative phosphorylation, we do have NADH plus the cytosolic NADH plus FADH2, which will be equivalent to 32 ATP, yielding a total net of ATP 36 ATPs. We will now continue with the anaerobic pathway. The anaerobic metabolism does not require the presence of oxygen to convert raw materials into energy. Anaerobic glycolysis is far less efficient, producing only 2 molecules of ATP in comparison to aerobic metabolism, 36 ATP. The body relies on anaerobic respiration when a sudden burst of energy is required in a short amount of time. For example, imagine that you are a sprinter or a weightlifter. Your physical demands are usually intense, but only for a limited amount of time. Since aerobic metabolism takes more time, the body uses anaerobic metabolism to generate energy for immediate use from carbohydrates, but not fat or protein. Beginning with a single molecule of glucose, the glycolysis process unfolds in the cytoplasm of a cell and does not require any organelles. In addition, for activities such as sprinting or weightlifting, the body is in short supply of oxygen. The heart is pumping blood as fast as it can, but not enough and not in time to satisfy the needs of the muscles or other cells. Unfortunately, one of the byproducts of anaerobic metabolism is lactic acid, which can cause fatigue. A rapid buildup of lactic acid is what causes cramps in athletes who push themselves too hard without properly warming up or when the body fails to balance aerobic and anaerobic metabolism. The body's default is aerobic metabolism since it is so much more efficient than anaerobic. Anaerobic respiration is there in case of emergency situations until the body can get back to its original oxygen-dependent self. So how carbohydrate can be converted into lipids? In most animals and in man, when dietary carbohydrates are in excess, the oxidation of a part of glucose maintains relatively high concentrations of ATP, NADH, and NADPH. When the maximum glycogen storage capacities are reached, the last two pathways are particularly favored, thus leading to the synthesis of lipids and especially triglycerides, which form an extremely convenient energy reserve. Now, the acetyl-CoA from pyruvate, which is the end product of glycolysis, will be turned into fatty acids through the fatty acid synthesis. The hydroxyacetone phosphate, that's the DHAP, a glycolysis intermediate, will be converted into glycerol 3-phosphate, which will then be added to fatty acids and undergo lipogenesis and be converted to triglycerides. And triglycerides are fat or lipid deposits in adipose tissues. Sugar turns into fat when an excess amount is consumed. While carbohydrates are the body's main energy source, it's important to eat the healthy complex variety and limit the unhealthy simple variety to avoid weight and fat gain. Fat tissue is actually responsible for converting excess carbohydrates into fatty cells. The process of conversion occurs when adipose or fat tissue forms a compound known as acetyl-CoA from glucose. I would like to conclude my report by giving examples of aerobic and anaerobic carbohydrates metabolism found in sports. In sport, we need energy, and ATP is the one giving us energy to do work. We have to sustain that energy to complete a training or a competition. The preferred energy pathway of athletes is dependent on the length of time they are competing and at the same time the type of exercise or sport they are into. One anaerobic energy system that is readily available for them to use as energy is the ATP-PC phosphocreatine energy system. It needs creatine kinase enzyme in the process in order to produce ATP. However, there is limited phosphocreatine in the muscles that lasts for only around 10 seconds of activities. This will be quickly utilized by the body and will require activities with short, quick bursts of energy to be efficient. Another anaerobic pathway in utilizing energy in sports is the lactic acid system. After ATP-PC has been used up, the muscle will now synthesize the glycogen through anaerobic glycolysis. Lactic acid will build up in the muscles and may indicate muscle fatigue or cramps for athletes working beyond their lactate threshold. This energy system will only last up to 30 seconds of fueling with 2 ATP produced. In this time, the athlete should either be slow down or stop his physical activities. For long-distance endurance athletes like in Ironman, Spartan race, marathon, etc., the preferred pathway of energy system is the aerobic energy system. Okay, we have discussed already the glycolysis process, not in producing ATP. But I would just like to take note that the preparation regimen for long-distance endurance athletes will not only be on storing and in storing fuel and good nutrition, but more so on skills training and conditioning, both physical and mental. The concept of energy continuum will tell us that at any given time, all the energy systems are in use. The proportion is determined by intensity of demand for energy. So as we can see, okay, during the 6 to 10 seconds until 30 seconds, we are actually utilizing the anaerobic energy system. And after that, we continue with the aerobic pathway or energy system. And if we go beyond, okay, more than our carbohydrates can supply, then the aerobic lipolysis or coming from the fat, energy will be the one to be utilized. In long-distance endurance sports, we want to keep our body energized for a long period of time. Thus, we need to maximize the energy reserves in our body or help our body to store energy for refueling. Want to try carbo loading? Thank you very much and God bless.